So unlike a regular classroom where things are methodically taught in a programmed way, when I'm tutoring someone on various subjects, I just try to ascertain where they are, and we kind of just trial and error find out what is known and unknown and what they've been exposed to. And so then I'll try to come up on the fly with what we're going to cover. So I'm joined here by Katie.
Hello.
Hello. And I'm going to be starting to teach her a little bit about bioinformatics. She's not heard the word FAST before. So everyone's in their specialty in biology. And there are a lot of things in school that are many of the traditional basics that aren't necessarily reflected in the cutting edge of professional biology. I'm not saying that it's a criticism of academia. It's just sometimes technology is moving so fast, and by the time it gets incorporated in the textbook, it's already obsolete because everything's moved on. So we're going to cover basic bioinformatics. And Katie, you let me know if I said a term you're not familiar with.
Okay.
So I'm going to share my screen. All right. Can you see it says human zinc finger?
Yeah.
Okay. They come up with all these funny names for proteins, and it's usually the researcher that comes up with it. So the way we can represent proteins, when you take biochemistry, you'll learn about this. You have 4 levels of structure for proteins, what they call the primary, secondary, tertiary, and quaternary structure. The primary structure of proteins is really just a sequence of amino acids that make up the protein, and they can be represented by English alphabetic characters. So like M would represent methionine, D for aspartic acid, I think, S for serine, V for valine. Can you see my pointer, by the way?
I can, yeah.
Okay. A is for alanine, F is for phenylalanine, E is for glutamic acid, D aspartic acid, et cetera, et cetera. And in fact, here we have it. Here is a little legend that actually explains it with the names, and you can correlate that to amino acids. Any questions?
What are the highlighted? Why are some parts of this highlighted?
Okay, so very good question. That was really important. So the way that it's spelled in a protein is it's just kind of like one long string. And you, for the benefit of the audience, can you state informally the central dogma of molecular biology?
Yeah, it's transcription, translation, and then proteins.
Very good. So the DNA, which has an alphabet of ACTG, the RNA has ACTU, so that's adenine. Oh goodness. A is for adenine, C is for cytosine, G is for guanine, T is for thymine, and U is for uracil. I used to sing this to my nephews to teach them the alphabet. They'd say, "Now I know my ABCs, next time won't you sing with me?" And then the amino acid alphabet, so DNA gets transcribed to RNA, RNA gets translated to proteins, and proteins have a different alphabet, which is very similar, which is basically the English alphabet. And it's one long string, but we can do a wraparound here. So you can kind of imagine that this is just one long string.
Mm-hmm.
But one thing that we will find is that the C's occur at regular intervals, and so do the H's. C is for cysteine, H for histidine. This is a very non-random pattern. So as you look at this, you could see there's already a level of organization here that's very clear. I'm going to show you that's very different from a collagen. This is collagen, and I highlighted the G's, the glycine amino acids. By the way, in biochemistry, they often call it residues instead of amino acids. I guess it was something, a residue is something left over. And in the early chemistry and the study of amino acids, they said, look at all these leftovers, and they just called them residues. So a term you might hear a lot is just hearing the word residue. I sometimes use it myself. These are glycine residues. Isn't that a striking pattern to you?
Yeah, it is.
Okay, so this gives it biophysical properties. And one thing that's going to be very telling is when you line the two up beside each other. So you can take this zinc finger, and I highlighted the C's here, and I'll show you the purpose of the C's and the H's. That's where the zinc ion actually connects.
Mm-hmm.
And you have to have the right spacing. Otherwise, I mean, you can tolerate a little deletion here. You know, it could be off a little bit, but you definitely have to have two C's and two H's. And I don't know if I have a picture of that. So why don't I just show it to you? Let's see if I have one here. Okay, here it is. So let me zoom this out a little bit. I hope you're getting the whole, sorry, a little disorganized. Okay, so we have a number of ways to represent a protein. One is just with the spelling. We have what we call Richardson ribbon diagrams, which actually shows you how the amino acids could be laid out. And then we have kind of an ad hoc one like here. This one's a nice conceptual diagram here. We have the zinc ion,
Mm-hmm.
and it connects to the C's, the cysteines, and then the histidines. So that's very important that you can kind of see why they have to be spaced appropriately. Because if this is too short or too long, it's not going to bind. The letters in between are very important. The zinc gets this to fold in a certain shape. And I'll explain a little later, maybe not in this discussion, but this is a very important, actually, since we just talked about transcription, zinc fingers, if you think about it, let me see if I have a picture. There was a pretty, yeah, this is a beautiful picture. It got its name because the zinc finger, when we talked about transcription, it's amazing. You actually need proteins to be able to go to the DNA so that the DNA can make proteins. It's kind of a chicken and egg paradox. But to find the right gene, there has to be an addressing system inside the cell. And so there are codes that identify a region and say, hey, this gene is, this is going to be a, do you have a favorite protein? Just name one if you have one. Otherwise, I'll come up with one.
Favorite protein? I don't know. I can't think of anything off the top of my head.
Okay. I'm going to say my favorite's topoisomerase. Topoisomerase. And we'll talk about that one. That's a really cool protein. So, but you have, okay, the current estimate, and as far as estimates go, could be wrong, probably likely is, but the best estimate we have is there are 20,000 protein coding genes in the humans, excluding what they call isoforms and alternative transcription. So just say 20,000 genes, but you have to be able to, the cell has to know, okay, it's time to make some topoisomerases. We have to find the address of the topoisomerase, and they're like 3.3 billion possible positions. Well, the zinc finger is able to, so there's kind of an address here. We call it a promoter region.
Mm-hmm.
And the zinc finger is able to find that, and it will help start all the machinery, zinc fingers. And so when we look at the zinc finger protein, so the zinc finger here, the letters in between the H's and C's help it correspond to find the right location in the genome. It's amazing. Uh, to get a really specific one, you need multiple zinc fingers, and they're kind of like an array. So oh, here's a good illustration. So you have the DNA here with the ACTG, and you have the zinc finger protein, and a zinc finger protein will. This is not exact, okay, but one inexact model is each zinc ion, each zinc finger defined by a zinc finger, what they call a domain, will attach to 3 DNA letters. Actually, it's 4 with 1 overlap, but it's roughly 3. And so you need multiple zinc fingers. And when you get the right array, you can specify an exact address. It could find exactly that location in the genome, which I think is, to me, that's really cool. It's like you have to get all the numbers right to get an address. You don't want to get your neighbor's mail by accident or someone else living across the street. It has to be also, you need similar precision inside the cell to do this. And so the zinc finger is kind of like a little mailman, you might say. Let me walk you through this one here. So each of these lines is an individual zinc finger domain. And so each line represents a different zinc finger. And in this one, there are 13 zinc fingers. So that's why it's called a zinc finger protein. It has 13 of these. And so each row is a multiple domain. And by the way, interrupt me if you have any questions.
Okay.
No questions?
No, I'm just trying to process it right now.
Okay, so one way to look at this is how it implements a reading. It's basically like a reader, you might say. The way it's going to go inside the cell nucleus of a eukaryotic cell, it's going to bounce around all locations. It literally almost might visit every address. And then when it sees its match, bing, it has a binding interaction, which is just amazing. We have, in the pharmaceutical industry, we're trying to create gene therapies. So we've tried to make designer zinc fingers, and we're having a hard time because we don't quite know how to change the letters here to get it to go to the locations we want. This is how difficult the problem is. It's more than just getting the right location. Because it's bouncing around, it could end up in the wrong location temporarily before it gets to the right one, and that's bad. Biology is so well tuned that it will just lock onto the right address. So given that each zinc finger attaches to 3 DNA molecules, if you want to identify a region like say 39 DNA molecules, you'd need 13 zinc fingers. And we actually happen to have one that can identify, I think, like 55 DNA. It's the transcription factor 3 alpha. And these processes are going on in the human body all the time. It's amazing. So one way that I look at it is the zinc finger is like a key. And it's looking for locks. And so this is not a very precise diagram. So you can look at it like that as a zinc finger, or you could look at it this loopy structure. And I think I had an illustration. So there it is, transcription factor. It's 1,120 nucleotides long. This is transcription factor 3A. I didn't mark it here, but that's what it is. And so this zinc finger will identify something that's exactly 125 nucleotides long.
Oh, wow.
Yeah, yes, wow. That was when the researchers who studied this, they also said, wow. They said this is very sophisticated. That it could target something that specifically. So here's an illustration of when a zinc finger protein will lock on to a specific region of DNA, and then it has other connection parts like the crab connection part.
Mm-hmm.
It has, if you see like how a car is put together, the parts have a lot of parts have multiple connections to all the other parts, and that's the same in a biological system. So the zinc finger is the first part that will locate the DNA, but then it has these parts that will get other complexes to connect to it, and these have to match too. So there's lots of interlocking parts that are shaped just right. And as we talked about off, backstage, the way the interlocking happens, let's see, this is page 48, I apologize. This is, by the way, for the viewers, this is Essential Cell Biology. You can get the first 80 pages for free, and it's totally worth it because it's free and you'll learn a lot. You can get it for free on Kindle. So the way that things interlock, it has to be shaped right, and then it has positive and negative charges that are perfectly located so that it can meet its match. So it's kind of, I call it like its soulmate. It's able to find its soulmate in a sea of what seems like a random chemical soup. It's able to, all these molecules are able to find all their partners. And it's an interesting question in biophysics because all these quote unquote Brownian motion is just shaking all these parts together. And it's just like taking a bunch of parts, putting it in a bag and shaking it, and then out comes a car. That's at the molecular level, you can do that. At the level of cars, you can't. But at the molecular level, it actually leverages all that shaking to get stuff to connect. So do you have any questions?
So, like looking at this diagram, so this is occurring during transcription?
It can. In this case, before transcription, sometimes they are actually, there's a lot of silencing or activation. And this may sound surprising. A lot of DNA regulation is actually silencing. It's like we don't need that. So the human DNA codes for 37 trillion cells. All of those cells are unique, and they're definitely cell types. So a blood cell is different from a bone cell, different from a liver cell, different from reproductive cells. Let's see, what else do we have? Skin cells, different from bone cells. Brain cells, different from cardiomyocytes, heart cells. What other cells can you think of? Skin. I mean, they're like 200 cell types.
Yeah.
Actually, we both took cell biology. We should know some cell types. Let's see let's muscle cells.
Yeah.
Let's see. Eye cells, the cells in the eye, and nerve cells. Oh my goodness, how could I forget nerve cells? Nerve cells are very different from bone cells.
Yeah.
So how do they differentiate? Well, there's all this regulation to do it, but a lot of it is like, well, the nerve cell will turn off a lot of stuff it doesn't need that a bone cell needs. So that's actually how a lot of differentiation has happened. That may sound amazing that, the totipotent cell. I don't know if they taught that in your cell biology class. I learned it in another class about developmental biology.
No, I don't think I've heard about that before.
Okay, so when let me just stop screen sharing for just a sec. So when when there's formed a zygote, it has all the information to create everything else in a human being, and that's a totipotent stem cell. Anyone in the comments section correct me. That means it's capable of differentiating into all those other kinds of cell types. The reverse is very difficult to happen unless you do some special chemical stuff that's not normal. Okay, so once a cell becomes a bone cell, it can't start making nerve cells, neurons, and then vice versa. However, if you have a totipotent cell, it can split and divide, and one of its descendants could become a nerve cell. One can become a bone cell, another skin cell, et cetera, et cetera. Does that make sense?
Yeah, I think I've heard you're you're talking about stem cells, kind of,
Right, right.
and
So, yeah,
so
right,
that's like the embryonic kind of stem cell that people use.
right, exactly. So I think the highest level, the one that contains everything, is totipotent, and
Hmm.
then there's some that are called pluripotent,
Yeah.
that aren't quite as they can differentiate into a lot of cells, but not as many as a totipotent. And then you
Okay.
have differentiated cells. I had we were learning how to reactivate um, old cells and make them youthful, and I that's another topic that was just so fascinating, because we're finding therapies that can heal people that
Hmm.
have damaged organs. We can regrow their body parts, and we don't have to use embryonic stem cells. We could use their own cells, take their cells, and regrow. Like I interviewed a cardiac heart surgeon, Robert Matheny, and he figured out how to regrow heart valves, because he said, we're taking this stuff that we make raincoats with and using that to make replacement valves. He said that's terrible stuff. It's the best we could do up until recently, and he figured out how to regrow heart valves. And
Wow.
these are like so young and youthful looking, and they work really well. There's some problems, obviously, but um, we can think about healing people that are burn victims or deep scarring.
Hmm.
And we're learning by accident. We learned by accident how to stimulate this. And that's where you get into the epigenetics. But speaking of which, so going back to, you were asking, is this how it starts transcription? And in this specific slide, this is not specifically transcription, but a form of regulation that, get this right, a form of regulation that involves either silencing or activation. So what happens is, when you study biochemistry and chromatin modifications, there are ways we can mark the DNAs and saying, hey, look, we're switching all of this off. So when the transcription machinery comes off, it's coming around. This is just signaling. Don't start making more of this. So this is what this is. Just as a FYI,
Hmm.
the particular, what's called a zinc finger, we found that it binds to endogenous retroviruses. They thought endogenous retroviruses are always junk. Sometimes some of it may be junk, not all of it. I actually think that endogenous retroviruses had a function, and some of them went rogue and started damaging the genome. But they actually have a very important function, because they're located near regulatory sites that control gene regulation.
Hmm.
And so, like, this crab zinc finger will park here, and it will recruit all this machinery, and then it'll start flicking all these switches saying on or off. It's really amazing. I'll teach you that someday. So you could see how complex this is. This is just insane. But this all began. I'm grateful for your curiosity, but this all began because we were looking at a zinc finger.
Hmm.
So now, um, so I committed to teaching you. Oh boy, we're already running out of time. I'm already out of time. So so we'll cover this in the next section. So collagen has a different spelling, and it has a totally different role than a zinc finger. So like parts in a car, each part has different roles. It has different shapes, different geometries, different functions, and um, you can get different shapes of proteins just by changing the spelling, which is amazing. And I don't know if I have a picture of. I don't have an. Let's see. Do I have an immediate picture of? No, unfortunately, I don't have an immediate picture. I do have a picture of what collagen. The shape of collagen is different than zinc fingers. Now, this is the same zinc finger, but I colorized even more regions just
Hmm.
to emphasize. You can see that they can't have any common ancestor. And I've asked an evolutionary biologist. He said, "Yeah, you're right, Sal. There's no common ancestor for all major protein families." mean, there's just like no similarity between them. So that's a take home. So what I've showed you, they call this representation of collagen or any protein when you're just spelling it. We call it the FASTA or FASTA. I use the word FASTA. I think the proper pronunciation is FASTA. I'd say FASTA format. So now you've learned a particular new term, the FASTA format. This is a primary. Okay, so the primary structure is the spelling of the protein. One way of representing it is with FASTA.
Okay.
FASTA format. Now, you'll see sometimes they'll use abbreviations that are different, and that's okay. You'll figure that out. But anyway, so now you've learned FASTA, and that's actually the basis of a lot of bioinformatics. So then once you've learned this, you'll start to learn other tools. I'm going to briefly, let me emphasize briefly. So just to look forward to where we'll go next time, there is, so the way we're able to find out like the spelling of proteins and all sorts of other things is with the Uniprot website. I think the proper pronunciation, which I'm not very persnickety yet about, is Uniprot. So there's the Uniprot website. So since we just looked at it, ZN zinc finger 136 human. So I'm just going to show you. So we have zinc finger protein. I was looking for 136. I know what I'll do. Hang on. I really want to let you go here. It had its name way up here. Oh, it was in my zinc finger slide. Sorry. Oh, yeah. See, there are these other abbreviations for the amino acids. Okay, so ZNF 136. So I'm going to go to the Uniprot website, and I'll say ZNF 136. Let's see if we'll find it. ZNF 136. There we go.
Okay.
And they renumbered it. Okay, so let's look. And here it is. So I'll just search on FASTA, which I couldn't find. Structure. So we're going to start going through this. Oh, see, you get the spelling here.
Oh, yeah.
Here in terms of amino acids, and we can download this. And it'll be there's the FASTA file right there. And you can see that I reformatted it to look like that. So MDSVAF. And let's see if we get the same spelling there. MDSVAF. See, okay, now you've learned.
Yeah.
Now you put this on your resume and impress people. You have some familiarity with the Uniprot website. You're going to get more familiar with it. And there are other tools like BLAST, but I'd say this is the most important. And this is also where you start to see where they identify the regions of nuclear localization. And just, I happen to know this since I've done the topoisomerase. So when you go to the Uniprot website, and then I'll let you go, you'll be able to identify the regions. And this is how you get the colorizing diagrams. They'll say, okay, these are the yellow. What was the yellow? The yellow are alpha helices. The red are turns. The green are beta strands, which you'll learn about in biochemistry 201. And then you can also colorize all the important regions. And then your project will be the nuclear localization signal. So you'll get to learn how to look at Uniprot. And sometimes if they happen to have data they might tell you that they identified the nuclear localization, which is really cool that they have all this data that thousands of scientists have labored for decades to find, and it's right there.
Yeah.
So tell me what you thought about, okay, so you saw the addressing system with zinc fingers. That does kind of remind me of nuclear localization. So how about in closing, Woody, why don't you just share with the audience what you thought about nuclear localization?
I think it's a really interesting process, and yeah, I just think that it's definitely something that's been like intentionally designed by a creator. I don't know, like you said before, like I don't know how it would have like evolved. It's so like structured and direct. It's like a mailing system, and the way that the DNA can get in and out of the cell through the importins and the exportins. Yeah, I just think that it's very like intentionally created.
Wow. Okay, I didn't expect that. That was totally unrehearsed. I would obviously agree. And then you also see the addressing system with the zinc finger proteins and DNA. And there are all these, there's so many addressing systems. I mean, if you think about it, how can the cell actually function if we didn't have an addressing system and transportation system to get one part to the right destination? That is just amazing. And like I said, a lot of it's done with Brownian motion. It's just kind of like shaking a bag, and it's just going to find its partner. So what we probably will do is just start going through UniProt and just trying to find out, we'll start learning how to identify regions of proteins. Every protein has its own kind of, some may not have nuclear localization signals. Some will, some won't. And some of them will have different regions. And we'll just study a few of them. This is okay. I'll teach you about post-translate. You'll learn UniProt post-translational modifications, and let's see. This was a very difficult experiment. I actually worked with a researcher who helped do some of this. Once you get into quaternary structure. These are all nice terms to put on your resume, by the way. You study quaternary structure.
Yeah.
You can actually, so UniProt will identify, and then also what they call, there's one other buzzword to go is protein data bank and X-ray crystallography. I mean, you'll be spooled up real fast on all these, but then, a lot of publishing in the papers is just looking stuff up. Once you learn the basics, you can actually have very high impact on furthering knowledge, because when you can depict some of this stuff graphically and teach it to others, they'll be amazed. As you're seeing, there's a lot of stuff that you've learned in school, which is great, and that you have the foundation to be able to share stuff that they don't necessarily teach, which is really interesting. Now you could actually show nuclear localization signal to other people and just let them say, wow, So in closing, I held you about 30 minutes over time, but if you have any questions, then we can go backstage. I'm going to end the recording, and we can have just a few minutes to say goodbye.
Okay.
Any closing remarks?
Not right now.
Okay. All right. I'm ending the recording. Katie, don't log off quite yet. So to the audience, I want to say thank you for joining us on this little tutorial session. Take care and God bless you.